Thanks very much to both speakers, and I guess we move over to the panel now.
That's right. So we'd like to, both Hilary and Stacey are joining us online, and we should be able to see them shortly. I want to also invite Dr. Benoit Daniel back on for the panel discussion. So, similar to the last session, for folks in the room, if you've got questions, please come up to one of the microphones. I know we've got almost 200 participants online, and we see some questions already coming through. So continue to put your questions forward. To get us started, the last session was pretty lively. Stacey, there were a number of questions in regards to the approach or a need for a randomized clinical trial for all settings. It'd be great to hear your thoughts on settings and targeted therapies where the randomized clinical trial approach may not be as necessary. And then specifically, when we are looking at randomized clinical trials, there was a lot of discussion in the previous session about these sample size expectations from the FDA.
Thank you. Good morning, or I guess good afternoon. It's morning for me. This is a very difficult question to give a one-size-fits-all type answer, because I think that's what we're striving for right now, is trying to have diversity in the development plans and having some flexibility in the development plans. The emphasis is that we've seen a lot of studies that have done simple 3+3 designs. They've had about 12 to 15 patients, and then they jump into 100 to 200 patients in their expansion cohorts. And eventually they may lead to a registration marketing application. And there's not really a lot of time in that big jump to allow for kind of refining the dose and identifying maybe the best dose to move forward. So one of the things that we're considering is to include the randomization in the dose expansion portion. So depending on how the trials are initially designed, the patient populations are going out, it might be feasible to identify 2 dosages to look at in a randomized design. These randomized trials don't necessarily need to be fully powered like you would for a randomized piece for a registration type trial, but really to give you a sense of the dose response, the exposure response, the tolerability information, the activity information. It's really to kind of give an insight into like fuller understanding these dosages. We do understand, we've seen studies that are coming in that are, for lack of a better term, all comers. It's multiple solid tumors, trying to figure out the best patient population that this drug may work in. It may be really hard then to jump into a dose like refinement period. So sometimes there is like a proof of concept or activity piece kind of in the middle. And then once they kind of started honing in on the particular tumor or particular disease state they're going after, then might doing their dosage optimization. Other times the trials start off very targeted. They know the disease they want to line of therapy they want to be in, or their drug is very targeted to a specific type of tumor. Then we're seeing moving from a dose escalation, dose finding piece into like a randomized dose finding. As far as the study numbers are, I don't think it's really easy to tell us like what those numbers are going to be looking like. I think there's a lot of flexibility in what those numbers are going to look like depending on the rarity of the tumor, the separation or fold change in the dosages that are being selected, and in the PK and the biomarkers. So I think that the key is just to have a very thought out plan that's very well justified and come in and have our conversation with the FDA and get our feedback.
So, good. I'm Philip Josephson, our European regulator. I'll ask you a follow-up question on that, if I may. With respect to different types of therapies, protein kinase inhibitors versus conventional monoclonal antibodies or toxin-bound antibodies, what's your thought about the primary research questions that this RCT would have? And is it correct to understand that you would see the RCT, the phase 2 RCT as sort of, shall we call it, the default option that would need to be justified not to be done? Is that more or less your thinking?
Yes, we are heavily emphasizing a randomized dose comparison. Like I said, it doesn't necessarily have to be a fully powered, fully maximized, fully developed comparison. But the reason that's kind of being our default setting is it's really hard to tease out the differences in activity, differences in safety, differences in PK and other aspects that we need to look at if you're not comparing the two dosages head to head. We've seen some of these early dose finding trials where we've tried to tease out the differences in the doses and the numbers are just too small. You're talking about 3 to 4 patients at each dose level potentially. So it's not very informative. We've seen, where the first or second dose level might show activity and then we don't have activity for a couple dosages and then we have activity later. And I think it's because a lot of times it's a heterogeneous population and limited number of patients in those trials, and the response rates may be low in some disease states at only 30-40%, if not lower. I think as far as the monoclonal antibodies versus the small molecule drugs, I think the biggest difference for us is really what dosages are being selected. I think for monoclonal antibodies, we almost never determine an MTD. Small molecule drugs, you sometimes may hit that wall and identify it. But I think as far as the dose escalation plan, the selection of the dosages, I think that's what's going to be driven by the type of the molecule, not necessarily the study design.
Great.
Hello.
Question here.
Yeah, it's Katrin Ropalla from CDDF. I have a question for you, Hilary, because you had such intensive discussions with patients. So when we started off the meeting today, we asked what are the main concerns with dose optimization, and we had 2, and one of the 2 was time. When you discussed the, your optimal model for how dose selection should be done or dose optimization should be done in the future, what were the concerns from patients with regard to potentially slowing down drug development? Were there any concerns, any trade-offs that they thought is important to make, or what was their general perspective on potentially getting approvals later based on needing to randomize? And then I have a second question, but that's for the FDA.
So to be fair, we had a handful of patients that were involved in these discussions. It was a large group of different stakeholders, so not only patients. But overall, we kind of walked away with the conversations, recognizing that while the phase 1s may take a little bit longer, the early phases where we're identifying those the optimal dose, the hope is that then that makes later phases shorter. And so overall development should ultimately not be changing because you're able to optimize the dose sooner. We think about some of these post-marketing commitments that we've seen, and that is just extending. I know the drugs are available and on the market, but it's extending our full understanding of safety and efficacy, as well as tolerability. So I think that the sooner we can do it, that ultimately lends itself towards a timeline that's not going to be overly adjusted.
Can I ask a second question? Thank you. And that's maybe both to you, Hilary and Stacy. It's about the optimization of the monotherapy dose, but then also most of the anti-cancer drugs are often given in combination. So if you have a drug that's mainly intended to be done or developed in combination, how much time should we spend on optimizing and finding the best monotherapy dose before we actually do randomization in combination?
I'll take that one first. So I think the combinations, there's a little bit to think about, like you said, whether or not the drug has activity on its own, whether or not, what late line of therapy you're looking at, whether or not you're looking at 2 investigations, you're looking at an investigational agent with a standard of care, or approved therapies. So I think there's a lot of factors that have to kind of go into that combination piece in thinking about it. I think ideally when moving into the combinations, whether it's 2 investigations or a new agent with a backbone standard of care approach, I think we're still just looking for the same kind of justification. It doesn't mean that we want you to start all over again, like go back to like redoing like the monotherapy dose finding. what we're looking for is just that the combination is justified, like the preclinical, the non-clinical data, any modeling and simulation, all of the available PK, PD, safety, efficacy data that you have available are kind of supporting the combination moving forward for the dose combination. And that's kind of what our approach has been. As far as, as the monotherapy dosing and moving forward, some individuals and programs have shown that the drug really didn't have a lot of activity, so it's really hard to kind of really identify that dose. But then you're looking kind of put into combination, kind of enhance both dose, either being additive or synergistic. And so I think all of those need to be thought forward. I think part of the combination too with the modeling and simulation needs to be just how those two drugs are being put together. Are they sequential? Are they simultaneous? How those dosing schedules are set up. And I think there just has to be some thought into the process, some justification in the process, some modeling and simulation to help support moving forward with the combination.
Yeah, I'll just add that we did have quite a few conversations about the need for combinations due to a lot of the anti-cancer therapies being given in combination. And, a lot of what Stacey said is what we kind of identified as really just having that clear data package and that connection back to, why you're choosing the dosages that you're choosing for both of the products that are included in the combination.
And I just want to plug into that the FDA is planning 2 public workshops on combination. Both are going to be co-sponsored with different organizations. The first one's going to be focused kind of on probably more of a high level of our approaches and our thinking and our rationale. And there'll be an open dialogue with, industry and academia and patient advocates and such. And the second one is going to be really focused on the quantitative methods that can help figure out the study designs, figure out the dosages to be selected for both the various components moving forward. So I think hopefully in the next, both of those will be in the fall. So I'm hoping after that, there'll be a little bit clearer picture on how to handle combinations and some new indications.
Yeah, I think those would be really helpful. I mean, I see some questions coming on just as sort of follow-ons to combination discussions, but the questions are around late development or later lines of therapy versus earlier lines, and how do you manage your combination treatments and dose finding in those populations. So I think those workshops will be really helpful. I think we've got another question here.
Hi, this is Burak from Merck, Germany. Thank you very much for the presentation. I have a question about PRO in dose escalation studies, not the expansion. So it was mentioned maybe a breadth of PROs will be collected, and that so of course it's better to have more data for better dose finding, but then it's adding patient burden, and it's mostly very late line patients. So how do you see this tension here, patient burden versus having more data?
Yeah, it's a very, very good question and something we grappled quite a bit with. I think that the patients that we've spoken with and some of what we've heard is that when they're on clinical trials and it's a push to a cell phone or a device, and so that's an important step too. Is it an EPRO or is it a paper-based one? But if you in an ideal setting, if you can get a push once or twice a week to provide some information about your symptoms. I think when patients understand the reason why, that was one of the key things that we discussed, was when patients understand the reason why they're filling out these surveys about PROs and the impact, and not just at the initial conversation about, informed consent and that kind of thing, but over time, continuing to remind the patient the value of filling out their PROs, it really does make a difference, and they do find that filling them out is helpful. I don't necessarily need to go down this route, but another key consideration is whether those PROs are being shared with the physician or not. And in a lot of cases, they likely are not. And so as long as the patient understands that, again, in that informed consent conversation, and when you're having conversations with them throughout their care, ensuring that they know that you're filling this information out, it's really critical for a clinical trial so that we can understand how this is going to impact patients in the future. But for your care, you should have conversations with your physician about the adverse events that you're experiencing.
So to Stacy. You note in your draft guidance there's a specific emphasis on late toxicity, and I wonder if you could explain a little bit more on your thinking of how dose ranging, phase 2 dose ranging randomized trials would address late toxicity, or is there a sense here that you'd actually sort of ultimately come to advocate several doses in phase 3 trials? Because I guess there's a point where you start wondering whether the phase 2 program would need to be a phase 3 program to address the questions, isn't it?
Thank you for the question. I think as far as late toxicity is concerned, I think what we were really trying to get at in that it's just kind of not just be looking at the DLT window, which may be like 21 or 28 days, because that's traditionally what has been done. Basically a cycle of chemotherapy, which we're still using those kind of arbitrary timelines now for cycles, right? Because we don't cycle oral chemotherapy or oral targeted drugs. They're just given daily continuously. So I think what we're trying to get away from is just looking at the toxicity within that first 21 days or the first month and using that to kind of make a decision on like whether or not you should go up to the next dose level. I think what we're trying to look at too is what the impact is on some of these lower grade toxicities that Hillary alluded to. Like some of the patients have, grade 2 diarrhea, and we've heard this over and over again, where grade 2 diarrhea is several stools a day that has a dramatic impact on the patient's ability to get out and have a decent quality of life and enjoy some of the activities they want to do, because they always have to know where the restroom is. And that can't be done in some of the activities that they're doing. And so I think when we talk about long-term, we're not necessarily talking about years out, although some of that may be a concern depending on the mechanism of the drug and the toxicity profile. But some of it is like just understanding what's happening like month 2 or month 3, where we're not necessarily considering it, that we're, the longer term inability to look at that. And some of what we see with that, we can get assessments from is also the dose mods, and not just like the percentage of patients who are getting dose reduced or dose interrupted, but when is it happening? Is it happening on week 1 and majority of patients are starting to need a dose reduction on week 1? Are we seeing these dose reductions at week, like 8 weeks, 9 weeks, because of the patients are starting to say, well, you know what, I really can't tolerate having diarrhea, for multiple days, multiple times a day for months on end. Like this just doesn't work. And so I think the term long-term is really not what we're used to seeing with chemotherapy. When we're long-term, we're talking about like 6 months to a year, or, after treatment is over with. And I'm not suggesting that those toxicities aren't important. We still need to understand like those long-term, but I think as far as trying to understand what dose to move forward, we need to be thinking not just in the first month.
Okay. Thanks, that clarifies.
So we've got another question here online, and this was raised earlier, and this is to the European regulators here as well. What interactions in regards to Project Optimus are taking place between the FDA and the EMA, or even national competent authorities, to ensure that there's a robust platform and guidance that's consistent to support global drug development?
Well, I can tell you right now, we do have frequent interactions with the other regulatory agencies. We do have something called a cluster call, which has multiple agencies on that have regular discussions that both the other regulatory agencies as well as the FDA can raise questions. We do have sometimes, for lack of a better term, like a joint review, like we'll be working on our review on our end, but the application or marketing application is being simultaneously reviewed by another regulatory agency. And then during those discussions, there's multiple conversations that happen. So there is some interaction. I don't believe there's a lot of interaction kind of like early on development, although the cluster call does sometimes focus on during development because different agencies have the drugs at different time points. As far as the dosage optimization, I think having some of our international representations at different meetings, such as this one, I think that's where the conversation's starting to happen.
Dr. Daniel, I don't know if you want to comment on any activities at the MHRA.
Yeah, as Stacey has highlighted, we do work with the MHRA, with the FDA through the Project Orbis initiative. So we try and assess, or we are assessing oncology products together, or soon after the FDA have assessed it. So there is that interaction. Even while we were at the CHMP, the meetings that Stacey has highlighted, we have had discussions with the FDA. I'd like to ask a question, if I may, specifically to Stacey. With regards to the dose optimization, there is some expectation that there'll be some modeling data to inform the doses that would need to be evaluated in seeking an optimized dose. So there are some initiatives which the FDA have, like the model-informed drug development, and also pathways in assessing these regulatory dynamic tools. So do you have anything more you can add on these two initiatives, like the model-informed drug development and fit-for-purpose initiative?
So as far as the modeling piece, I think what we're trying to get away, I think with oncology, a lot of times we tend to look at the exposure response, the dose response, or even the population PK kind of very late in development. It's kind of like registration's done, all the data is available, let's go ahead and do those assessments. And I think what we're trying to do is encourage stakeholders and sponsors to kind of look at, covariate effects on exposure response, dose response, population PK as early as feasible based on their rich sampling that they've done and sparse sampling in some of the earlier trials, incorporating non-clinical data when it's relevant into those models. So that's what we're encouraging. It's kind of trying to push all the tools that we have, earlier in development to kind of inform dosing as sooner rather than later. As far as the meetings are concerned, we do have the model to inform drug development. We are starting to see that being implemented more, where companies are coming in with specific questions on their modeling and whether or not, the approach is reasonable or something that FDA is willing to take a look at. And I think those conversations have been very informative to the sponsors that we've had those conversations with. I don't have experience with the other meeting types. I know they're available. We're just starting to see the type D meetings as well, which we're starting to see those which have been focused on dosing and some specific questions. So I think the bottom line is the FDA is available to, meet with you, with stakeholders when there's questions key about dose selection. And we actually encourage it. We suggest after the first dose finding, dose expansion, we suggest that sponsors come in with a full data package. We have a list of things that we like them to include. They certainly don't have to include them all. And those are happening. We're also incorporating something into Project Breakthrough, which is the breakthrough designation to also try and get more to kind of help refine the dosing now that the breakthrough therapies done, make sure that that drug has an opportunity to optimize dose and move forward, even at the pace that wanted for a breakthrough therapy. So I think there's a lot of possibilities. There's in the Project Breakthrough, I think it's called the dosing toolkit is what it's called, I think officially. The name got changed a few times, but Hillary's nodding at me, so I know I got it right. But dosing toolkit, which is a list of kind of key questions or key things for sponsors to think about. It's meant to be a living document for those who are participating in this pilot project, and we're trying to just see how things are moving forward, trying to get information up front and getting some of these drugs developed.
Yeah, I'll just highlight that document that Stacey is discussing, one of the original versions of it is in our white papers as an appendix. And so it's a series of questions that just helps to guide that came out of some of our working groups focused on understanding how breakthrough has impacted and helped drug development in some of the ways that we could improve it moving forward. One of the key things was understanding drug development and dosing specifically.
Okay.
Perhaps a question to both of you. Perhaps the first part to Stacey, the second to Hillary. Traditionally, drug development has been extremely attractive to do that in the US. Now, for some biotechs, the time to the next, let's say, investment round may simply be too long. So they may need to think about different approaches and come with leaner packages. Any thoughts from the FDA how to deal with these? And then the second one, second part on this, perhaps same medal, different side of the coin. If pressure is usually the driving force, external pressures, the driving force for creativity, but you need to fund it. Are there any activities on the Friends side to think about the life of biotechs, where this creativity usually takes place before it's in-licensed by Big Pharma? These companies do not necessarily have the funds to run all these things that sound absolutely right and are definitely the right thing to do, but without money, it's a dream. What are the thoughts around this? So sorry for a long question. I hope I made it clear in the differentiation.
I guess I'll go first and then I'll pass it over to Hillary. So honestly, I think what all of us are stressing is just investing the time upfront will actually ultimately not really change may lengthen the initial part of the dose, of the drug development, but really shouldn't prolong the drug development. We're not trying to make the process more arduous or longer or decrease or dramatically increasing the time until these drugs get to market. What we're trying to do is make sure when the drug hits the market that the dose is optimized to the best of our ability. And optimized, is really hard to tell you what an optimized dose means. I mean, we're basically looking at the balance of benefit and risk. And so I think, those are some of the things that we're just kind of considering in the development process. But with that, I'll turn it over to Hillary.
Yeah, so this is a great question and something, again, that we grappled with quite a bit in our working group, because we did have, some representation from biotech, but it was largely with larger pharma companies. I do think that these earlier phase studies have the potential not only to understand dosing more effectively, but like Stacey said, shorten the overall timeline. That doesn't help a biotech company that's only really doing that early phase, right? And so I think two big things that came out of our most recent working group were understanding how to actually implement these studies. What do these studies look like now if we're going to start adding PROs on top of what we've already collected? And then the interpretation. So how are you as an organization and FDA going to interpret those results to identify what the optimal dosage is? And so those two pieces is where we're moving next. And one of the key things that we're thinking about is as technology continues to advance, what are some of the newer that we can be incorporating into the dosage escalation and the two studies for dosage optimization that can maybe replace, maybe improve, maybe enhance some of the ways that we're identifying or collecting data already. I think that the key piece of this is that it's not only about how do we collect better and newer data, it's also let's think about how that data fits into the overall paradigm. And so while yes, it might seem like it's a lot of information now, it's kind of balancing those two pieces, the older data and the newer data, to identify the most optimal data to inform what dosage you're using. And so I think, again, it can sound like it's a lot more, but if you can take away some of the data that we've already collected that might not be as important, I think that'll be a key piece of this.
Okay, I'll get back to the question of the randomized dose ranging trial. We're talking about pre-authorization here, but for many drugs, the first initial authorization is, let's say it's the late line use in a given disease, and then in the life cycle of the drug, the drug moves to front line, where obviously tolerability may differ from the later line, and you might also, it's not uncommon that we move to different diseases where not only tolerability, but also the exposure response may differ from the original indication. How do you see, is this dose ranging procedure, do you foresee that this will be repeated for each sort of extension of the indication that for a product, or will you, what's your thought on that?
So this was briefly touched on in the very end of the guidance document. I think it was probably the last bullet point on the last page. But the thought process is that no, we don't want each development for, I'm losing my terms, but we don't want each development program, like for each indication, for new combinations, for new disease states, earlier line of therapy, all of those switches to kind of go back and start all over again, because that's kind of wasting resources. It's wasting patience, it's wasting time, it's wasting a lot of things. what we're asking for is that data package to come in and justify the dose that's moving forward, because moving from later line to earlier in the line of therapy, you may have different mutations, which different responses to the different dosages, like different concentrations may be needed to actually affect that target. You may have patients earlier on who are treatment naive and don't have a lot of underlying toxicities because they haven't seen multiple drugs. So they may be able to tolerate a slightly higher dose to get the bang for the buck that they needed to get their, their cure, or get them to that point where they may be in, a sustained response for some period of time. So I think the bottom line is all of those factors need to be thought about, like moving from later line, earlier line, difference in genomic, mutations or resistance that are underlying, what their prior therapy has looked like, what their tolerability might be like. But I think all of those pieces, excuse me, need to be brought together to have an informed data package. And that data package may be simply like looking at the prior clinical data. It also may, if the first trial was a lot of all comers, multiple tumor types, but then the company focused on only one tumor type, but they still have some original data on some of the other tumor types that may help inform similar in dose response or exposure response. So I think it's just a matter of looking what information is available to you and understanding the disease state, understanding the patient population you're in, understanding what your drug has already told you. So the answer is no, we don't expect in most cases to go back and start all over again. We're just kind of a thoughtful process of pulling everything together to make a reasonable data package to justify that dose in the next population.
I've got one more question here, and again, for Hilary. In terms of Project Optimus, what is its applicability to pediatric developments? The draft guidance didn't go into much detail, so pediatric development is obviously at the forefront. And this question really is trying to pin down what are the expectations from the FDA.
I can't speak for the FDA, but I will say I'd echo what Dr. Ratine said earlier, just that, if you have enough patients to understand how a drug is working in a certain population, then you probably have enough patients to do a dose optimization study. So I would encourage, sponsors, again, not from an FDA perspective, but from a personal perspective, to just consider, have conversations with FDA and learn more about it. But I think it's probably a good idea to understand the pediatric population in a dose context.
So the only thing that I want to add on to that is it depends on where we're starting with pediatrics. Are we starting with pediatrics because it's a pediatric disease and we're starting de novo drug development, which we've seen a few drugs develop that way, where you need to do the dose finding in the pediatric population to figure out what dose you're moving forward? Or are you extrapolating from the adult data that you already have into adolescents where the dose is most likely the same? Or are you trying to extrapolate into a younger patient population where you may have to consider PK or PT differences just based on where they're at in their overall development? So I think, again, it's just, data package. It's just pulling all the pieces together, what you understand about your patient population, your disease state, how your drug is working, what initial clinical and non-clinical data you have to kind of just justify the dose that you want to be moving forward. So.
Yeah, I think we've reached our time, but thank you, Hilary and Stacey, for a very open discussion. I'm sure there'll be more questions to follow, but this has been very, very informative. Thank you very much.
Indeed. Thanks very much.